Hey guys, so this video is about the cell membrane. Often when we've heard about the cell membrane before AP Biology, people talk just about the lipid bilayer. I'm here to tell you there's way more going on in the cell membrane than just the phospholipids. We know that the phospholipids are there. We know that they form a bilayer, so there's two layers, and you can see in this picture that they're opposite from each other. So here's one layer, here's two layers. There's also proteins, and there's also other accessories. So the phospholipids are the main barrier. They allow small molecules through, and they're amphipathic, which means they have a hydrophobic end and a hydrophilic end. So an end that likes water and an end that does not like water. The proteins are for transport, so small molecules can get through the phospholipids, but if they're bigger or if they're charged, they need a protein to be able to go through. And there's accessories, so for example, there's cholesterol, glycoproteins, and glycolipids, and I'll talk a little bit more about what each of those are as I go through the video. So people refer to the plasma membrane, the cell membrane, as a fluid mosaic model. So the reason for that term is fluid because the cell membrane is fluid. It's not a solid, it's not rigid, it can move. And mosaic because it's a mosaic or combination of things. It's not just phospholipids. You can see there's lots of stuff in this picture, so it's a fluid mosaic. In this picture, you can see our phospholipid bilayer, so the purple circles with their tails, phosphate group on the head, and lipid tails, just like you saw in the POGIL that you maybe already did, hopefully already did. So that's the phospholipids. We also can see the proteins. There's lots of different types of proteins in the cell membrane that are embedded right in there, or sometimes they're kind of on the side. They're peripheral proteins. There's also glycoproteins and glycolipids. So if we think about what those words mean, glyco is anything that has to do with a sugar, and protein, we know that means protein. So glycoprotein must be a sugar attached to a protein. And in fact, this is a glycoprotein right here. So there's a protein that's represented with blue and a carbohydrate group, so a sugar group coming off the top. So it's a glycoprotein. Glycolipid, again, sugar, lipid. Lipid is fat, so it's a sugar attached to a lipid or a fat. So if we look at this one, you can see hopefully that it's attached to purple. So it's attached to a lipid. That's what I'm trying to represent here. And not to a protein. So this is a glycolipid. It's a carbohydrate attached to a lipid. This is a glycoprotein, carbohydrate, sugar attached to a protein. Another thing that we have in this picture is cholesterol. So you've probably heard that word cholesterol before in terms of either you should eat cholesterol or you shouldn't eat cholesterol. Your body makes some cholesterol, and some cholesterol is good for us, and it resides in the membrane as well, and I'll talk a little bit about what that is for in a little bit later in the video. Some other things to keep in mind in terms of the structure of the membrane. So the outside are the phosphate groups, the phosphoheads of the phospholipids. They are hydrophilic, meaning they like water, hydro, water, philic, like, while the inside or the tails is hydrophobic. Okay, so this inside in-between layer is hydrophobic. It does not like water, but the outsides, either side where the purple is, the top and the bottom, those are both hydrophilic. They like water. In this picture, I'm showing the outside of the cell as this top part, and the inside of the cell is the bottom part, and there's a reason that my glycolipid and my glycoprotein have the sugar group or the carbohydrate group sticking out on the outside of the cell. Okay, so the proteins and lipids and everything else in the membrane move around a lot. So like I said, it's not a solid. All of this stuff is constantly moving and shifting and flowing around. An experiment that scientists did to figure that out that I think is really cool is they took two different cells. One cell they labeled with one type of protein. Another cell they labeled with another type of protein. So proteins they could detect, they could see when they looked at the cells. They put those cells together so that they merged together into one cell. And then when they examined that cell after an hour or some set time period, they could see that those proteins had distributed all around the cell. So I don't know if you can see colors on this or not. But in this cell, in this mixed cell, there's purple and red, and they're spread out everywhere. It's not still on one half of the side of the cell or another half of the cell. Another important thing with the fact that it's a fluid, oh, crud. Okay, still talking about cell membrane structure. When the lights turned off, I was talking about the fact that it's important that the cell membrane is a fluid. And you know how butter is a lipid, right? It's a fat that we know about. If it's in the refrigerator, it's a solid. If you held it in your hand, it starts to get softer and can turn into a liquid if you warm it up. So if you are outside in the cold, your cells with their lipids in them, they want to start to become a solid, just like butter becomes a solid when it's cold, if it were outside or if it was in the refrigerator. So that would be bad for your cells if your cells had a solid membrane. The proteins and other things in there wouldn't be able to move around. Your cells wouldn't be able to have things coming through the cell membrane because the phospholipids would be packed too tight and it would be too rigid. And some cells wouldn't be able to get around in your bloodstream. So the way that your cells combat that is they change what type of phospholipid is in the cell membrane depending on the temperature. So for example, in a standard phospholipid bilayer that we have seen before, we typically draw those lipids relatively straight. We draw them kind of squiggly because that's what carbon chains are. They kind of go back and forth in real life. That's their structure. As it gets colder, your cell is going to incorporate more unsaturated fats into the phospholipids. So we learned already that an unsaturated fat, that means that it doesn't have hydrogens at all the spaces that it could have hydrogens. So you can see here this one doesn't have hydrogens there. And what that does is it kind of puts a kink into the fat. So you can see in this picture here how the unsaturated phospholipids kind of have a bend. So this one here that's a little bit yellow, that's an unsaturated lipid. So it puts an extra space in there. So what that does is as the cell membrane starts to cool off and it wants to be a solid, it's preventing those phospholipids from packing so closely together so they can't turn quite into a solid. So the colder you are, the more unsaturated fat is in your cell membrane. The warmer you are, the more saturated fat, the more straight phospholipids, because they're not going to be as attracted to each other or get as stuck together if they're warm. They're going to be able to move past each other pretty well. Cholesterol is also something in your cell membrane that is there for that purpose. So as you can see in this picture, the cholesterols in green also kind of have a kink to them. They kind of go off to the side. They're not straight. They also prevent the phospholipids from packing closely to each other and let the cell membrane continue to be fluid even if it cools down. Okay, so we talked about the phospholipid bilayer, and we talked about the accessories, the cholesterol, the unsaturated fats, etc. I'm going to talk a little bit about the different types of proteins. See if we can get rid of these lines. Maybe not. Okay, I think we're just going to have to deal with those lines. So there's lots of different types of proteins in your cell membrane. There are transport proteins. Some require ATP, some do not. There's proteins that are enzymes. They're catalyzing reactions that happen next to the membrane. There are proteins that send signals, receive and send signals. So if this is the outside of the cell and there's these orange colored pi signals floating around out here, when one lands in the receptor of this protein, the signal would be sent down throughout the cell to where it needs to go, that signal is present. There's also cell-to-cell recognition. So this is where glycoproteins come into play. The carbohydrate chain on the end is often specific to certain types of cells. So if another cell comes along, it can detect that specific structure and know, oh, hi, you're my friend, or hi, you're not my friend. There's also intercellular junctions. So if cells need to stay really closely next to each other, so like in your small intestine, where you don't want the stuff that's going through your intestine to just be able to flow into your bloodstream, you want that barrier, so there's a filter. The cells are connected really close with each other through an intercellular junction. And there's also proteins that their job is attachment, so maybe to hang on to something in the extracellular matrix, stuff outside of the cell, or to hold on to the cytoskeleton, something in the cell. So their job is attachment. So I also want to take a step back and give us a chance to remember where these proteins are coming from. So we learned that the DNA lives in the nucleus, it's copied into mRNA, mRNA leaves the nucleus, goes to the endoplasmic reticulum or a free-floating ribosome, and there the mRNA is made into protein. So in this case, I'm going to try to zoom in on our endoplasmic reticulum. So you can see here, this is supposed to be my membrane protein right here, this blue. So our ribosome made that membrane protein that's going to rest in the outside of the cell membrane once it gets there. And a glyco group or a carbohydrate group is attached, that green little ring that you can see there. So from the ER, the membrane protein stays in a membrane, so this membrane breaks off of the ER, goes to the Golgi. We can follow it through the Golgi. In the Golgi, the carbohydrate is modified a little bit. I don't know if you can see that there's like a goal post here. So instead of it being a U, the Golgi modified that carbohydrate group into a different shape. It keeps going, keeps going, keeps going, and eventually this membrane is sent to the edge of the cell and becomes part of the cell membrane. So as you can see, the glyco part of the glycoprotein is on the outside of the cell. Dang it. All right, guys. Okay, so that light is not going to turn off again because Miss Lay is here and she's going to wave at it to keep it alive so we can keep talking. All right, so where I left off was we just heard about a protein that was made in the endoplasmic reticulum, got sent through the Golgi to the outside of the cell to be a cell membrane protein. I just wanted to point out that throughout the process, the glyco part of the glycoprotein was inside of the Golgi, inside of the endoplasmic reticulum, inside of the vesicle. So that's the part that ends up outside of the cell, so you can kind of watch it happen. So in summary, what we talked about in these now 3 videos, that was supposed to be 1 video, was the membrane is made up of phospholipids, proteins, glycolipids, glycoproteins. So many different things, not just phospholipids. And also it's a fluid. It moves around. It's not a solid. Okay, thanks guys.